Welcome to the deep dive. Today we're tackling a really foundational topic, looking right at the genome's architecture.
Yeah, we're going deep into Chapter 6, variation in chromosome number and structure.
Our mission, as always, is a solid, college-level review. We want to cover the key ideas, the discoveries, and how these changes in chromosomes actually, affect organisms.
Exactly. We're focusing on cytogenetics, that blend of cell biology and heredity. And these variations, they're not just theory. They're behind genetic disorders, big evolutionary leaps, and even, well, how we grow our food.
Speaking of food, let's kick off with something maybe a bit surprising. Bread wheat. Triticomostivum, it's basically a genetic superstar built on, well, complexity.
Oh, absolutely. It's maybe the perfect case study for how rearranging genomes can be incredibly successful. Modern wheat, it's a hexaploid.
Hexaploid meaning?
6 complete sets of chromosomes, so 42 total. It came about through a series of hybridizations, weaving together genomes from 3 different ancestral grass species.
Wow. So it wasn't like a single clean event. It was messy, step by step.
Pretty much.
Yeah.
Hybridization, then chromosome doubling, then maybe another hybridization, another doubling. And this whole process, this triple hybrid nature, it gave wheat fantastic traits.
Like bigger grains, right?
Yeah, and Brazilians.
Exactly. Larger grains, ability to grow in way more places than its ancestors. It just highlights perfectly how changes in chromosome number and structure aren't always bad. Sometimes they're the engine of, well, progress.
Okay, so if we want to study these changes, these successes, and sometimes the problems they cause, we actually need to see the chromosomes. How do scientists do that? What are the tools for cytogenetics?
Right. It's a pretty careful process. First step, you need cells that are dividing quickly. White blood cells are often the go-to grown in culture.
Okay, dividing cells. Then what?
Then you need to trap them right when the chromosomes look best. That's usually metaphase, when they're most condensed. You use a chemical, colchicine is a common one, to mess up the mitotic spindle.
So it freezes them mid-division?
Essentially, yes.
No.
Arrests them in metaphase. But even then, for a long time, counting was tough because they were all jumbled up. Yeah, I remember reading the human count was thought to be 48 for quite a while. What fixed that?
A surprisingly simple but brilliant technique. Treating the cells with a hypotonic solution.
Hypotonic. Low salt.
Low solute concentration, yeah. So water rushes into the cells via osmosis. They swell up, puff up like little balloons.
Spreading the chromosomes out inside.
Exactly. Then when you fix them and put them on a slide, they spread out beautifully. That single step was crucial for getting the accurate count of 46 human chromosomes. Made it precise.
Okay, so they're spread out. Now we need to tell them apart. The old stains just colored everything uniformly, right? How do the modern ones work?
We use differential staining now. Quinacrine was an early one. It's fluorescent, binds to DNA, and creates these unique bright and dark bands under UV light for each chromosome pair. Very reproducible.
And the other main one, Giemsa.
Giemsa stain, yeah. That's probably more common now. It's not fluorescent. You usually treat the chromosomes with trypsin first. That's an enzyme. Digest some proteins, and then stain with Giemsa. This creates the classic G-banding patterns, really distinct for each chromosome.
Those bands are like the address on the chromosome, right?
Yeah.
But what if you're looking for something really small, like a tiny change or a rearrangement?
Good question. That's where you need more advanced techniques, like chromosome painting or IFI-esh fluorescence in situ hybridization.
How does that work?
You create these DNA fragments called probes, and you label them with fluorescent molecules. These probes are designed to stick, to hybridize, only to specific matching sequences on the chromosome.
So you can light up a whole chromosome, or just a tiny piece.
Both. You can paint an entire chromosome one color, or use different colored probes for different segments. It makes it incredibly easy to spot things like translocations, where bits have swapped between chromosomes or complex rearrangements. They just jump out visually.
That visual map leads to the karyotype, right?
Yeah.
The standard picture.
Exactly. You take a photo of the stained chromosomes from one cell, digitally cut them out, pair up the homologs, the matching pairs, and arrange them by size, largest to smallest, plus the sex chromosomes. For humans, that's 46. 22 pairs of autosomes and the XX or XY sex chromosomes.
And this karyotype lets us talk about specific locations precisely. The P and Q arms.
Right. Standard nomenclature. The short arm is P think petite, and the long arm is Q. Then we number regions out from the centromere and subregions within those. So something like 5P sub 5.3 tells you exactly. Chromosome 5, short arm, region 25, subregion 3.
And that level of detail matters because even small changes can have big effects.
Huge effects. That's why having this precise map is critical.
Okay, so we have the map. Now let's talk about the variations themselves. You mentioned changes in number and structure. What are the main categories?
Broadly, you've got the normal state, which is euploidy having the correct complete sets. Then the variations fall into two big groups. Polyploidy, which is having extra entire sets of chromosomes.
Like the hexaploid wheat 6N.
Exactly. Or triploid, 3N, tetraploid, 4N, and so on. The other big category is aneuploidy. That's an imbalance in just part of the genome, usually gaining or losing a single chromosome.
Let's stick with polyploidy first. You see it all over the plant kingdom, right?
Yeah.
Potatoes, bananas, cotton, half of all plant genera.
Yeah, it's incredibly common in plants, often leads to bigger cells, and thus bigger, more robust plants. Think larger fruits, flowers. Great for agriculture.
But it's super rare in animals, especially ones that reproduce sexually. Why the difference?
It mostly comes down to sex determination. In many animals, especially mammals like us, having extra sets of sex chromosomes, like XXX or XXYY, completely disrupts the balance needed for proper development and sex determination. It's usually lethal very early on, or leads to sterility if the organism even survives.
Okay, that makes sense. You also mentioned sterility. Why are many polyploids, like triploids, 3N, often sterile, like seedless bananas?
It's a problem during meiosis. Meiosis needs pairs, right? Homologous chromosomes need to pair up precisely before they separate into gametes. If you have 3 copies of each chromosome, how do they pair? It gets messy. Very messy. You might get one left out a univalent or all 3 trying to pair up a trivalent. When the cell divides in meiosis the chromosomes segregate randomly, so the resulting gametes get, an uneven number. Some might get 2 copies of a chromosome, some might get 1.
Yeah, aneuploid.
Exactly. Wildly aneuploid. And those unbalanced gametes are almost always non-viable. That's why things like seedless bananas or watermelons have to be propagated asexually using cuttings or runners.
But wait, our wheat example was fertile. You called it an allopolyploid. How does that work?
That's the key difference. Allopolyploids arise from hybridization between different species. Let's say species A, genome AA, hybridizes with species B, genome BB. The initial hybrid might be sterile, AB, but if chromosome doubling occurs...
You get AABB?
Precisely. Now you have a tetraploid, AABB. During meiosis in this plant, the A chromosomes have perfect partners, other A chromosomes, and the Bs pair with Bs. Segregation is usually normal, producing balanced AB gametes, so fertility is restored. Wheat is even more complex, involving 3 species, but the principle is the same.
Before we move to aneuploidy, what about polyploidy within an organism? Endomitosis. And those giant fly chromosomes.
Right, it's not always the whole organism. Some specific tissues can become polyploid through endomitosis DNA replication without cell division. You see this in human liver cells, for instance.
And the Drosophila ones, polytene chromosomes.
Oh, they're spectacular. In the salivary glands of fruit fly larvae, the chromosomes replicate over and over, maybe 500 to 1,000 times, but the strands stay stuck together, forming these incredibly thick banded cables.
You can see the bands under a light microscope.
Easily. And what's really cool is, first, the homologous polytene chromosomes pair up tightly, band for band. Second, all the centromere regions clump together in one spot called the chromocenter. They were, and still are, invaluable for mapping genes.
Okay, fascinating detour. Let's shift now to aneuploidy, the gain or loss of individual chromosomes. This messes with gene dosage, right?
Absolutely. Gene dosage is critical. Having 3 copies of the genes on 1 chromosome instead of 2, or only 1 copy instead of 2, throws off the balance of gene products.
There was that classic study with Giemsa weed, Datura.
Yes, Blakesley and Belling back in the early 20th century. Giemsa weed has 12 pairs of chromosomes. They found 12 different mutant lines, and each one had a unique, distinctively shaped seed capsule. Turned out each line was trisomic, had 3 copies for 1 specific chromosome out of the 12.
So trisomy for chromosome 1 gave one shape, trisomy for chromosome 2 gave another, and so on.
Exactly. It was a beautiful demonstration that having an extra copy of just 1 chromosome changes the organism's development in a specific way because of the altered dosage of genes on that specific chromosome.
In humans, the most well-known aneuploidy is trisomy 21, Down syndrome.
Right. 47 chromosomes total, with an extra copy of chromosome 21. The effects include characteristic facial features, developmental delays, increased risk of certain medical issues like heart defects, and earlier onset of Alzheimer's-like symptoms.
And this usually comes from prenatal.
Meiotic non-disjunction. That's when chromosomes fail to separate properly during meiosis, either meiosis the first or meiosis the second. So one gamete ends up with 2 copies of chromosome 21, and another gets none. If that gamete with 2 copies is fertilized, you get trisomy 21.
Is it linked to parental age?
Strongly linked to advanced maternal age. Female egg cells start meiosis before birth, but then arrest and prophesy for years, even decades. The thinking is that over time, the protein complexes holding homologous chromosomes together might degrade slightly, increasing the chance they won't separate correctly when meiosis resumes later in life.
Most other autosomal trisomies are lethal, right?
Hmm.
But sex chromosome aneuploidies can be viable, like XXX or Klinefelter.
Yes. Things like 47XX Triploids syndrome or 47XXY Klinefelter syndrome are viable. The reason they're tolerated much better than autosomal trisomies is X inactivation.
The process that shuts down extra X chromosomes.
Exactly. In cells with more than one X, all but one get randomly inactivated early in development, becoming condensed bar bodies. This helps balance the dosage of X-linked genes, making the aneuploidy less severe. Although there are still often some effects, like infertility and Klinefelter syndrome.
What about losing a chromosome? Monosomy.
Autosomal monosomies are generally lethal very early. The only viable human monosomy is Turner syndrome, which is 45X.
So only one sex chromosome at X.
Correct. These individuals are female, but typically have characteristic features like short stature, a webbed neck, and are infertile because their ovaries don't develop properly. It also arises from non-disjunction or sometimes the loss of a sex chromosome during the first few cell divisions after fertilization. This can lead to mosaicism, where some cells are 45X and others are normal 46XX.
Okay, that covers the number changes. Let's switch to the final category, structural variations. Rearrangements. The pieces are all there, mostly, but they're rearranged.
Exactly. Bits of chromosomes get deleted, duplicated, flipped around, or swapped.
Let's start with deletions and duplications, segmental changes.
A dilution, sometimes called a deficiency, is just what it sounds like. A piece of a chromosome is missing. The individual is hypoploid, has less than the normal amount for the genes in that segment. A well-known human example is cri-du-chat syndrome.
Cri-du-chat.
Right. Caused by a deletion on the short arm of chromosome 5, written as 5p. Affected infants often have a distinctive cat-like cry, plus other developmental issues.
The duplications.
The opposite. A segment is present in extra copies. The individual is hyperploid for those genes. The classic example is the bar eye mutation in Drosophila.
Fruit flies again.
Yep. Flies normally have round eyes. The bar mutation causes the eyes to become narrow, slit-like, or bar-shaped. It turns out this is caused by a tandem duplication. The segment is repeated right next to the original of a specific region on the X chromosome, region 16A.
And the more copies, the worse the effect.
Exactly. Flies with one duplication have bar eyes. Flies engineered to have two duplications have even narrower, ultra-bar eyes. It's a perfect example of how gene dosage from a structural change directly impacts the phenotype.
Okay, next. Inversions. A piece flips around.
Right. A segment breaks off, rotates 180 degrees, and then reattaches. The key distinction is whether the inverted segment includes the centromere.
Paracentric versus pericentric.
Yes. Paracentric means beside the centromere, the breakpoints are both in one arm, so the centromere isn't included in the flip. Arm lengths don't change. Pericentric means around the centromere. The breaks are in opposite arms, so the centromere is part of the inverted segment. This can change the relative lengths of the P and Q arms.
What happens when these try to pair in meiosis, if someone has one normal and one inverted chromosome?
The heterozygote. To get the homologous sequences lined up properly, the chromosomes have to form this characteristic inversion loop. You can actually see these loops under the microscope in things like those polytene chromosomes.
Looks complicated. Does it cause problems?
It can, especially with crossing over within the loop. For paracentric inversions, crossover can lead to chromosomes that are broken or have two centromeres, usually resulting in non-viable gametes. For pericentric, crossover can lead to gametes with duplications and deletions, so inversions can reduce fertility.
Okay, loops for inversions. What about translocations? Swapping pieces between different chromosomes.
Right. Reciprocal translocations involve an exchange of segments between two non-homologous chromosomes. Imagine chromosome A swaps a piece with chromosome B.
If you inherit one normal set, A, B, and one translocated set, A translocated, B translocated, how do they pair?
It gets even more complex. To maximize pairing, all four chromosomes come together at their homologous regions, forming a cross-shaped structure, or a cruciform, during meiosis the first.
A four-way junction. How does that separate properly?
Well, that's the problem. There are three main ways that cross-shape can segregate. Only one way, called alternate segregation, produces balanced euploid gametes. One with the normal A and B, one with both translocated versions.
And the other ways, adjacent I and adjacent II.
Both of those lead to unbalanced gametes. They end up with duplications of some segments and deletions of others, essentially partial trisomies and partial monosomies. These are usually inviable. So individuals carrying a reciprocal translocation often experience significantly reduced fertility, maybe around 50% reduction, because half their gametes are non-functional.
Okay, one last category, fusions. Compound chromosomes and Robertsonian translocations.
Compound chromosomes are fusions involving homologous chromosomes, like the attached X chromosome sometimes found in Drosophila, where two X chromosomes are fused at the centromere.
And Robertsonian. That sounds specific.
It is. It's a specific type of translocation involving two non-homologous acrocentric chromosomes.
Acrocentric. Centromere near the end, tiny short arms.
Exactly. In a Robertsonian translocation, two acrocentric chromosomes fuse at or very near their centromeres, forming one larger chromosome. Often, the tiny short arms are lost in the process, but they usually contain non-essential genes, so it's often viable.
And this type of fusion is particularly relevant to us.
Hugely relevant. If you compare the human karyotype to that of chimpanzees, gorillas, orangutans, you'll see they have 48 chromosomes while we have 46.
Where did two go?
They didn't disappear. It's strongly believed that human chromosome 2, which is a large metacentric chromosome, centromere in the middle, is the result of a Robertsonian fusion event that occurred in our ancestral lineage after we split from other apes. It fused two smaller acrocentric chromosomes that are still separate in apes.
Wow. So a single chromosome fusion event marks a major step in our own evolution.
It seems so. A major structural rearrangement distinguishing our genome.
Okay, we have covered a tremendous amount today. From seeing chromosomes, to counting them, to understanding how extra sets or single extras affect things, and finally how rearranging the pieces drives change.
Yeah, we started with the cytological tools like staining and swelling cells. Then we hit numerical variations. Polyploidy, so important in plants, and aneuploidy, the cause of syndromes like Downs and Turner's due to gene dosage issues. And finally, the structural rearrangements. Deletions like in cri-du-chat, duplications like Bari, those inversion loops, and the fertility consequences and evolutionary significance of translocations, including that big Robertsonian fusion in our past.
It really emphasizes that the genome isn't static. These rearrangements, deletions, duplications, fusions, they're constantly reshaping things. We started with wheat, this complex hexaploid that evolved over maybe 10,000 years through hybridization and doubling, and we ended with human chromosome 2, maybe formed by one Robertsonian event. Makes you wonder, doesn't it? What other major genomic restructurings driven by these same processes are happening right now in other species, setting the stage for future evolutionary paths we can't even predict?
That's the core question, isn't it? How these structural changes fuel the engine of evolution. It's an ongoing process.
Absolutely fascinating stuff. Well, thank you for walking us through that intricate world of chromosome variation. And to you, our listeners, thank you for joining us on this deep dive. We hope this review helps solidify these crucial concepts for you. We'll catch you on the next one.